This is Bio 181, Chapter 12 on genetics. And now we're moving into non-Mendelian genetics. So these are genetics that have been discovered more recently than those originally formulated by Mendel. So they're exceptions to pure dominant and recessive patterns. So we'll start with variations on dominance. So dominance is not always complete. And actually that arises again and again in this section. So the two that we're going to look at here are incomplete dominance and codominance. Okay. So the examples that are shown over here on the right are with two parents. We've got one parent that has red flower color. So we're going to be talking about capital R. Oh, yeah. Let me just use black. It's going to be easier. So capital R is for red. Okay. And then W is going to be for white. Okay. And notice that I'm not using capital and lowercase. They're both capital. So they're codominant or incomplete dominant. So there's no recessive in this case. So they're both capital. So in this case, for incomplete dominance, we have one parent that's red. So their genotype would be RR if we had the original type of notation. And then the white in this case would be WW. Okay. But what we're doing is, because we are using a new framework, we're actually going to write these as a superscript. So for the red, we're going to put it on top. So I'm going to write it over here. So the red flower is C superscript R, C superscript R. So C is, in this case, we're just going to say C is flower color. Okay. So we're representing the gene, right? And then R is the red allele. So R, red. I wrote it up there, but W is white. Okay. Right. So the genotype of the red flower is C superscript capital R, C superscript capital R. So that's the formation, the notation we're going to use. Okay. So this is the red flower. Now we're going to cross that. We're going to breed it with the white flower. And so again, we're going to use C for flower color, and then superscript above it, we're going to use capital W for white. So this is the white flower, white flower. And then you're just going to use the same Punnett square. I'm going to do it down here in the middle. Okay. So here's the Punnett square. Put a box here so it's less distracting. So we're going to do the red flower. I'll put it over here on the left side. So it's going to have two red alleles. Okay. So this is the red parent. And then up here on the top, we're going to do the white flower, two white alleles. Okay. And then we're going to combine them in the four boxes that represent the four possible offspring. We'll combine the superscripts as well. Okay. All right. All right, so in this case, we have 100% of the offspring are heterozygous, so they have one of each allele. In the case of incomplete dominance, did I put my color thing? It disappeared. Shoot. Oh, there it is. So lost it. Okay, so here it is. So in incomplete dominance, let's find the pink. The heterozygote is going to have a phenotype that is intermediate to the, that's hard to kind of see, but that's pink. It's a dark pink. You could get a darker pink. Okay, darker pink, quick, darker pink. All right, that's whatever. So over here, this genotype in incomplete dominance is going to be intermediate. So it's like a blending of the red and the white. So in this case, it's pink, right? And this is the case of incomplete dominance, incomplete. Okay. So 100% of the offspring, if this trait is inherited with an inheritance pattern of incomplete dominance, then all of the offspring would be pink. If flower color is inherited in a pattern that is codominant, then 100% of the offspring are actually going to have both of the traits expressed. So the CRCW is going to represent like patchy. So both traits expressed, both red and white color petals, okay? And both of these are found in nature. So some plants have pink if they have intermediate phenotypes for incomplete dominance. If it's codominant, then it's going to be over here, codominant, dominant. Oh my God, dominant. Okay, all right. That's enough for this one. Next slide. Multiple alleles. So sometimes you have more than two alleles. This is called multiple alleles. For example, you know, if you have a cat, meow, right? So cats may have, for their coat color, different colors. And so you might have black, which we could represent with a B, and you might have orange for the coat or the fur, which would be O, and you might have white. So these are all possibilities. Now, any given cat is only going to have two of these because cats are diploid. That means they have, you know, they have a gene that codes for fur color. Okay, so here's my fur color, right? So any given cat is only going to have two alleles. So it could have an allele for black and an allele for orange, or it could have an allele for black and an allele for white, or it could have an allele for orange and an allele for white. So you get a lot more possibilities when you have multiple alleles. Okay. An example of this that you should know is the ABO blood group. So your blood type is determined by multiple alleles. You could have the allele for A, you could have the allele for B, or you could have an allele O, okay? So if you have a genotype of AA, okay, or AO, okay, you are type A blood. If you have the genotype that is BB or BO, you have type B blood. B. If you have one A and one B, then you are type AB, right? Type AB blood. If you have both O's, two alleles that are both O, then you are type O blood. Type O. So what does this mean? What are these pictures on here? it turns out that your red blood cells have proteins sticking out of them, right? So we talked about that a lot. So you've got a red blood cell, here's the plasma membrane. And out of the plasma membrane, there are proteins, identity markers, right? So there are these proteins that stick out and they have markers on them, okay, that identify them as a certain type of cell. So one of the identity markers is called A, and then on red blood cell, you can have another type of identity marker. Okay, don't mess the colors up too much here, which would be look a little bit different. Okay, so it looks different, but slightly different sequence of maybe a glycoprotein B here, right? So if you're a type A cell, you only have the A's, right? So you only have, well, that's supposed to be blue. So you only have the A's. So here you only have this one type of protein of interest, and that's, I'm trying to draw this quickly, right? If you're type B, you only have this type of protein. Oh, gosh, okay. You only have this type of protein. So over here in your red blood cell, right, that's the only thing that's sticking off. If you have AB blood type, you've got both, right? You've got both types. So that's actually another example of codominance within multiple alleles. They're both expressed at the same time. So that's codominant. And then if you're type O blood, you don't have either of those. Okay. So that is blood type. The last thing for this set of this video is epistasis. So epistasis is where we have interactions between multiple genes. So you've got, say, a gene here on chromosome 1, right? And then maybe over here on chromosome number 10, right? We've got 23 pairs of chromosomes. So here's 1 and here's 10. So here's a gene that we're going to talk about on chromosome 1, and maybe it's for fur color. And then you've got another gene over here on chromosome 10, and this is for pigment deposition. Pigment deposition, which just means you're making the molecules themselves for that color, deposition. So together, these can interact to change the phenotype. So let's look at an example. I'm just going to go, I'm going to draw it all out. I've got an example here. So this isn't a mouse with epistasis. This is from our book. It's an example. And so we've got capital A in this case is for brown fur. Lowercase a is for black fur. Okay. And then we've got another gene, which is gene C. So both of these are for, this is for fur color, right? So that might be on chromosome 1, right? And then I don't know which number it's actually on in the mouse, but in the mouse, then you've got this pigment deposition, right? That's going to be on another chromosome. Let's just say it's on 10. We're going to call that gene C. And so if you have capital C, that's normal production, right? And if it's a lowercase c, it doesn't work. You have no pigment. Okay, no pigment is produced. It's defective. So that means anytime you have any of these genotypes that have capital C's in them, they're normal. This is the normal type. Whoops, that's not. Yeah, that's normal. So any of these that have the capital C in them, they're all normal because they're all making pigment. Where it gets weird the interaction occurs is if you have two lowercase c's, you have the two recessive alleles for no pigment. That's down here. Let me just use a different color. So let's use red. Okay. So down here, all of these mice that are white have the genotype of homozygous recessive for the pigmentation gene. Okay. So the interaction between the pigment gene and the fur color gene is an example of epistasis. And then you get a ratio, a phenotypic ratio here at the bottom of the brown or agouti mouse, right, is 9 out of 16. The black mouse is 3 out of 16, and the albino mouse with no pigmentation is 4 out of 16. All right, that's it for this video.